Okay, so this video is going to cover Hardy-Weinberg Equilibrium. This is a tool to measure the expected amount of individuals in a population. So like any number of animals, plants, you could say, pretty much whatever you want to measure. We can measure the frequency of the genetics and the phenotypes and the genotypes of species, at least what we would expect when they meet these conditions. So these conditions basically are saying, like, here's the expectation if there's basically no genetic variation at all. No migration, no mutations. There's I believe there's random mating. There's a couple others that we'll cover in class. What you need to know is that this is essentially just, these are the expectations if no gene flow is happening and this was just like a random population of things. So to start, we can imagine kind of a small like plant population, let's say. So we covered these in the genetic section. These are incomplete dominant alleles. So in that case, here you have a red flower. This is a pink because it's 1 and 1, so it's blended. And this is white. So we're going to measure the presence of each of these alleles in a population, and we can measure the genotypes as well. So let's say that we observe just random numbers of population species, or of a population. In this case, the total of this population that each of these genotypes is 47. So the frequencies, what this is, are just the frequencies of each of these genotypes. So all that is, if I ever ask you for a genotype frequency, that's just the percentage or decimal, whatever one of you want to do, of each type of genotype out of the total. So 15 divided by 47, that is 31.9%. And you could do decimals too if you want. It's probably easier, but I'm just going to do percent. 25 divided by 47, 55.6. And then 7 divided by 47, that's just 15.6%. Okay? So those are your genotype frequencies. Those are all figured out just if we already know those. So that's easy, so that's good. So if I want to know the frequency of each of these alleles, that's kind of the next step. So if I want to just see, let's say, to start, if I want to know the frequency of the R allele, let's say that, so since we have 47 individuals, each of these has 2 chromosomes. That means that our total number of alleles is 2 times 47, which equals, just let me be sure, 94 alleles. And I also have my notes, just chromosomes. So we're kind of counting chromosomes here, even though we're just counting genes. So our total alleles is 94. So the number of R alleles can be calculated kind of as follows. You have 15 individuals with double R. So that means that there's 2 times 15, because each of these individuals has 2 of these. Okay, so plus, though, these 25, because of these 25 individuals, they each contribute 1 R. So that's 25. So what this works out to be is 55 of all the chromosomes are a dominant allele chromosome. That's out of 94. And that's going to equal 58.5%. So that's the frequency of R in this case. So this R dominant allele. So we'll see that we can do this in normal Mendelian genes. I just think it's easier to treat each allele the same to start. So remember, these are both, this is incomplete dominance, so every cell is kind of 50-50 if you get a heterozygote. So we can do the longhand for the W allele. So there's 7 individuals with 2 each. Okay, so that'll end up being 14. Plus 25 with just 1 each. So that's 25 again. And in this case, we equal 39 out of 94. And you can see, as I'm slowly doing my notes for my math, these will equal each other. So the way that we write this is, in this case, the frequency of R plus the frequency of W is always going to equal 1.0. And that's if we're doing decimals. So in this case, 100%, same thing. So how you're normally going to see this written is P, which is dominant allele, and Q, which is the recessive. So in this case, we have 2 dominants. It's the same thing though. They equal 1. So that means if you actually just know the frequency, let's say of W, and so the P and the Q, this is just algebra, we just have to use these to show. But let's say that we're using our example. We know that the R frequency plus the W frequency allele, that's going to equal 100%. So if we know, for example, that the R allele is this 58.5%, we know that the W allele, based on this algebra, is going to be 0.415. So that's a good, easy way to kind of remember that, is that the allele frequencies themselves are always going to be equal to 1. So we have these numbers now. We know that R is 58.5%, W is this frequent in the population. Okay, so we'll erase this. So from this population that we found, we know that this is the genotype frequency of everything, and we know that we can find the allele distribution. Okay, so let's use this for one last thing.